Hello, my name is Dr. Matt Piercy. I'm a faculty member at Yavapai College. I'm building a series of videos that talk about the biology and the illness caused by the COVID-19 coronavirus. One of the topics in an upcoming video is going to be the vaccines that are currently in development to help inoculate people against infection by the virus. Before I build those though, what I thought I would do is build a video first that talks about how vaccines work. So what I want to do first to talk about this is play this video that you can see that was made by the University of Oxford. This does a really good job of explaining how vaccines function. And then after that, I want to talk about 3 different ways that you can actually go about building a virus. And then after this video, then we can go on to talk about the different vaccines that are currently being developed in an attempt to inoculate people against infection by the virus.
To understand how vaccines work, it helps to look first at how the immune system works, because vaccines harness the natural activity of your immune system. There are about 100 trillion bacteria and viruses on your body. Not all of them cause disease, but some are able to get inside our bodies to multiply, and this can make us ill. There are barriers to stop this happening, but suppose some disease-causing bacteria do get through. Your immune system is quick to recognize them as invaders. This is because the proteins or sugars on the bacteria's surface have different shapes to any of the ones in the human body. They trigger a complex chain of events involving many different types of white blood cells working together. One type of white blood cell is able to make antibodies to fight the invaders. Antibodies can stick to the proteins or sugars on the bacteria's surface, and this kills the bacteria or disables them. However, not all antibodies will work against these bacteria. They have to be exactly the right shape, a bit like a key fitting a lock. Our bodies have a library of billions of white blood cells, each of which can make just one shape of antibody. Only a few of these antibodies will match the invading bacteria. Producing antibodies of the right shape can take several days. By this time, there could be billions of disease-causing bacteria in your body. Once the right cells are activated, they quickly divide and turn into a production line, making masses of antibodies that stick to the bacteria. Eventually, your body gets rid of all the bacteria and you recover. Antibodies remain in the blood, and some white blood cells may also become memory cells. If those specific bacteria invade the body again, the immune system will respond so quickly that you won't get ill. Vaccines work in the same way. They contain weakened or dead bacteria or viruses, or even just a few proteins or sugars from the surface. This is enough to convince the immune system that a real invader has got in. The same process takes place as when real bacteria or viruses invade our bodies, except you don't get ill. Afterwards, if your body ever meets the real thing, your immune system will remember it and get rid of it before you even know it's there.
So I think this video does a really good job of explaining the advantages of vaccines. So first, when we're infected by a virus or a bacteria naturally, it can take up to a couple weeks to get high levels of antibodies. So in that time, we can get very, very sick or even die from the infection. So what a vaccine does is teach our bodies how to make antibodies to a particular virus or bacteria. And so if we're ever actually infected with the real bacteria or the real virus, our body already knows how to make antibodies to that microbe. So it doesn't have to go through that two-week period of learning how to make them. So a lot of times, if you've been vaccinated for something, you may actually be infected with that organism or that microbe and never actually know it, because your immune system, having already learned how to fight that particular microbe, makes antibodies to it so fast and destroys it so quickly that you never even know that you're infected. So when we want to make a vaccine, the ultimate goal is to teach the body how to fight the microbe, but do it in such a way that the person never actually has to get sick. So there's 3 different ways that we can do that are shown on this slide here. So the first thing we can do is vaccinate someone with what's called an attenuated vaccine. In an attenuated vaccine, what we do is we take a virus or a bacteria that's been weakened. It can still replicate, but it does so very slow and very weakly. So as long as you have a healthy immune system, that organism can't make you sick. But when we inject someone with that weakened organism, their immune system still finds it, recognizes it, and make antibodies to it. But the person doesn't get sick because the bacteria or the virus is weakened. And, months later, years later, hopefully, if that person's ever infected with the actual wild type or normal microbe, then their body already knows how to make antibodies to it. And so they can fight that microbe off and probably never get sick. This is how we make a vaccine to measles. We can also make what's called an inactivated vaccine. In an inactivated vaccine, what we do is we take a bunch of the microbe and we inactivate it. So we basically kill it, but the chunks, what we call antigens in immunology, or pieces of that microbe are still around. So we inject that mixture, basically debris from the microbe, into somebody. That's what the vaccination is. But your immune system still finds those parts, those antigens of the microbe, and mounts an immune response to them and makes antibodies to those parts, that debris, those antigens. And then later on, if you're ever infected with the actual organism, because your body knows how to recognize the parts of the microbe, then it can recognize the intact microbe as well and make antibodies to it very quickly. This is how we make some of the vaccines for polio. The third way we can make vaccines that's shown right here is by making what's called a subunit vaccine. In a subunit vaccine, what we do is just in a lab, we will use molecular biology techniques to make just a single part of the microbe. So the whole microbe isn't there. It's just a piece of it. It's a piece of the microbe or an antigen that we know our immune system can find based on previous research. So what we do is we make a bunch of those subunits or a bunch of those antigens or parts of the microbe, and then we inject that into somebody. Because the whole microbe isn't there, the person is never actually ever going to get infected with anything, but their immune system is going to learn how to fight that one small part, that subunit or that antigen of the microbe. And then if the real microbe ever shows up and infects the person, because the person can make antibodies to that subunit, that part of the microbe, then of course it can do it to the entire microbe should that person ever be infected. So those are three ways that we can teach the immune system how to recognize and how to fight a microbe by making antibodies to it without the person ever becoming ill. So in future videos, we're going to talk about how we're harnessing these techniques to build vaccines and test them for the COVID-19 coronavirus.